If you look closely at any living thing, a blade of grass, a butterfly's wing, the tip of your finger, you'll see delicate lines, intricate patterns with scales, valleys and ridges creating even more patterns. But if you look closer and deeper with a microscope, you will see a common structure that makes up every living thing: cells. Let's take a closer look at them. Get your paper ready, wide right, skinny left. One invention, the microscope, changed biology so profoundly, as much as the automobile changed transportation. Let's take a look at how this invention changed my subject. In the mid-1600s, as we talked about earlier in the year, Anton von Leeuwenhoek saw animalcules. He saw tiny living creatures that he thought were living molecules. Not long after the invention of the microscope, Robert Hooke was observing a slice of cork and noted that it seemed to be made of thousands of empty chambers. He called these chambers cells because they reminded him of the cells or tiny rooms in a monastery. Today we know that the cells are not empty, but they contain living matter, and they function like a microsystem that keeps all living things alive. It also became very obvious that cells were the basic unit of life. Discoveries were being made and confirmed at a fast pace. From these discoveries came the concept of cell theory. Cell theory has three basic parts. They're pretty common sense now. All living things are composed of cells. The cell is the basic unit of structure and function. And new cells are produced from old cells or existing cells. As the Hubble telescope allows us to see further and further out into space, millions of light years away, so do advancements in microscope technology allow us to see further and further into things, seeing details we never imagined. From the light reflected by a mirror on the earliest microscopes to directed light microscopes like we have in our classroom here, to transmitting electron or scanning electron and now scanning probe microscopes, microscopes have taken us on a fantastic voyage. Just as with Hubble in outer space, we can see details that were unimaginable by early biologists. Take a look at the surface of this snowflake. The image of the iris of a human eye. Or this avian flu virus. These microscopes allow scientists to discover exciting new worlds and to learn more about how life is constructed. One important discovery with these awesome microscopes was that there are actually two types of cells: prokaryotes, or cells without a true nucleus, and eukaryotes, or cells with a true nucleus. Life as we know it consists mostly of eukaryotic cells, but electron microscopes have shown that bacteria and viruses are prokaryotic. Knowing this allows researchers to effectively work on more ways to cure illnesses and perhaps eliminate the viruses or bacteria that cause them. Pretty cool. Organelles are little organs that work together to help a cell function efficiently. So they're kind of like the departments in a factory or an office. They really do work together like the organs in your body. We're going to focus on the eukaryotic cell in this unit. There are two main parts to most eukaryotic cells: the nucleus, as you see here in the brown, and the cytoplasm, which is everything outside the nucleus but inside the cell membrane, including any organelles. The cytosol is the liquid portion of the cytoplasm that suspends all the organelles. So let's take a look at the inside of the cell. We'll start in the very center and move out. So first let's look at the nucleus. It's the control center of the cell. It contains DNA and coded instructions for making proteins and other important molecules. While most pictures show the nucleus in the center of the cell, it can be anywhere in the cell at all, or not present at all, as in red blood cells. The nuclear envelope, as the name implies, surrounds the nucleus. It's filled with pores, making it selectively permeable. It allows materials like proteins in and out of the nucleus, like messages and instructions moving in and out of the boss's office. Within the nucleus are three very important structures. Chromatin is a granular material made of DNA bound to proteins. It condenses to form chromosomes. Chromosomes are thread-like structures that contain all the genetic information that is passed from generation to generation, and it ensures that a species can continue. And then there is the nucleolus. This structure is actually inside the nucleus. This structure is a little like a spot on an auto assembly line where the chassis of a car is put on the line. Without that spot, there would be no car. It's here that the all-important ribosome assembly begins. One of the most important jobs of this cellular factory is making proteins. Proteins are assembled on ribosomes. And remember, ribosomes were made in the nucleolus. Ribosomes themselves are small particles of RNA and protein, and they make more protein following coded instructions from the nucleus. Cells involved in protein synthesis are packed with ribosomes. Which now leads us to my favorite part of the cell, the endoplasmic reticulum. The ER is an internal membrane system, a pipeline between the nuclear envelope and the cytoplasm. It's here that lipid components of cell membranes are assembled, as well as proteins and other materials that are going to be exported from the cell. And there are two parts to the ER, endoplasmic reticulum. The rough ER has ribosomes stuck to it. Remember what ribosomes do? They're involved in protein synthesis. They make protein. And the rough ER chemically modifies proteins coming from the ribosomes. The smooth ER does not have ribosomes stuck to it. It's involved in lipid synthesis and the detoxification of drugs. Liver cells contain large amounts of smooth endoplasmic reticulum. As we continue to follow this assembly line, we move on to the Golgi apparatus, named after its discoverer, Camillo Golgi. Easily identifiable in a cell, looking like what I think is a stack of pancakes and located next to the ER, proteins move from the endoplasmic reticulum to the Golgi apparatus to be modified, sorted, and packaged, either for storage or secretion outside the cell. The Golgi apparatus is like a customization shop, putting the finishing touches on proteins before they leave the factory. Lysosomes are the cleanup crew of the factory. They are organelles filled with enzymes that break down and digest lipids, carbohydrates, and proteins into usable molecules. They also break down old organelles that are no longer useful. Our tour inside the cell next moves to the vacuoles. These are sac-like structures that are used for storage of materials such as water, salts, and proteins. Plant cells have a single central vacuole filled with a water-based liquid that helps maintain pressure within the plant to support structures like leaves and flowers. In animal cells, vacuoles are important in maintaining homeostasis. Food can be found in a vacuole, and it's digested by the cell, and wastes are released all within the vacuole. You see that happening here along the bottom of this cell. In other cells, vacuoles act to pump water around and out of the cell to maintain the water balance within the cell. Factories are hooked up to a local power company for a source of energy. Cells have the mitochondria. These are present in almost all eukaryotic cells. They convert chemical energy from the digested food into usable energy. The mitochondria is characterized by an outer membrane and an inner folded membrane. Another interesting fact about the mitochondria is that it comes from the cytoplasm of the ovum or the egg. So you inherited your cytoplasm in your mitochondria from your mom. If you ever read about DNA testing, you often see referrals to mitochondrial DNA, and that provides links specifically through the maternal or mother lines. Chloroplasts are found only in plant cells. They contain chlorophyll and they capture sunlight, converting it to heat energy and allowing the cell to convert water and carbon dioxide into a chemical known as carbohydrates. We see the chloroplasts as the green little dots in these plant cells. Think about what life would be like if chloroplasts did not exist at all. Pretty scary. Just as steel or cement beams support the walls in the factory, the cytoskeleton is the support structure of the cell. There are two types of filaments, both of them made of protein, found in the cytoskeleton. Microfilaments are thread-like and they support and help with the movement of a cell. Microtubules are hollow structures that help maintain a cell's shape. They also form centrioles that are important in cell division. More on that later in this unit. Microtubules also help to build cilia and flagella, which allow single-celled organisms to move around in water. In the diagram here, you see microfilaments shown in the bottom left, and you see the microtubule shown towards the top right in the blue. Both of these, as we said, help with support and movement within a cell. Our journey through the cell leads us to the last two structures. The first is the cell membrane. All cells have one. The cell membrane regulates what goes into and out of the cell. It also provides support and protection. It's composed of a lipid bilayer with protein molecules embedded in it and carbohydrates attached to the protein molecules, as you see in this picture. So once again, we're hearing about those carbon compounds. Plant cells have cell walls also. Cell walls lie outside the cell membrane, as you see here in this picture. If you look up in the top left corner, you'll see the cell membrane to the inside and the cell wall to the outside. Cell walls also provide additional support and protection for the plant. They are composed of a very tough carbohydrate called cellulose. In fact, this substance is so tough, our digestive system can't digest it. Diffusion and osmosis are the two main processes that occur at the cell membrane and the cell wall. Remember that diffusion is the movement of materials from high concentration to low, like tea in hot water. And osmosis is the diffusion of water across a selectively permeable membrane, like a cell membrane. The goal of both of these processes is always equilibrium, or the equal concentration of materials throughout the cell. These cell parts and processes are vital then to homeostasis. So, some questions to think about while you are chewing on plant material. What is cell theory and what's it all about? What cell parts are involved in production or breakdown or boundaries? And how do they work? You should also be able to talk about what might happen if a cell part doesn't function correctly. And lastly, Why are plant cells different than animal cells?